Welcome to Chapter 21. This chapter covers the miscellaneous bacteria of disease, and so they're under the category of miscellaneous because they are either atypical bacteria or they have a very unique shape, such as the Spirochetes. There are some curviform bacteria, such as Campylobacter jejuni, which causes GI distress, and so these being varied, multiple shapes, things like that, make them miscellaneous in category, since the others were gram-positive coccus, gram-positive bacillus, gram-negative bacillus, etc. So spirochetes are a very unique bacteria. The two families of spirochetes that we're going to discuss are that of Treponema and Borrelia. So they are gram-negative. They are free-living, so these guys can be in water, soil, and things like that. They also can be commensals, meaning that they can live on the host. The bacteria benefits, but the host does not. Or then, of course, the ones that are primary pathogens causing harm, such as Treponema. Treponema pallidum causes syphilis. We are indeed the natural host. This bacteria is described as being fastidious, which means that it's very high maintenance, does not survive outside of the body for very long periods of time, and so it is transmitted through sexual contact because you have to have those close associations for this particular bacteria to go from person to person. So in that sort of scenario, you can't acquire syphilis from sharing beverages, utensils, things like that. And so one of the most renowned cases of syphilis was that of the Tuskegee Institute. In 1932, there was a study where they wanted to further understand syphilis, the disease mechanisms, and kind of come up with how does this disease progress in an effort to eventually treat it. Well, as they're going through this study, they broke down a group with men with syphilis, so those were the test group. Men without syphilis, those were the control group. And so they were offered health care, food, meals, burial stipends, rides to and from the clinic, and they were given some minimal health care provided simply by if you have a cold or things like that. So they treated anything except the syphilis. They did want to see syphilis in all stages. And so the controversy arose really in 1947 when penicillin was shown to be a complete cure for syphilis. And so this research study had been going on for some time, and it actually progressed all the way until 1972, so another 40 years from the start of the trial, where patients' families have been suffering from syphilis, because syphilis, being a sexually transmitted infection, is not only going to impact the patient, but it's going to impact any children that are born. It's going to impact their partners. All of those people then are going to be infected with syphilis simply by the nature of the disease. And so this study, when it came to light in 1972, it became pretty much the prototypical case of failure to exude ethics in a scientific setting. And so there's a $10 million settlement that is still being paid out today to the participants, their wives, their children, all those family members, as a consequence of this grave injustice. So the first stage of syphilis is that of primary syphilis, and I've given you a whole variety of places where you could find a canker. So cankers can be found in the mouth, in a tongue, finger, on the actual genitals. And so this canker is very, very contagious. So the canker has a high quantity of spirochetes at that particular sore site. And so if you have a canker and you are sexually active, then you will then transmit syphilis to your partner. So the deal is, this canker will actually resolve in about 3 to 6 weeks. It will spontaneously heal, and at this point the spirochete is moving from the epithelium into the blood. Once the spirochete is multiplying in the bloodstream, we now call this secondary syphilis. It is characterized by a rash. Shown here are images of a patient with the rash on their hands, rash on their chest or back. And so the rash appears and then disappears. So you ultimately would not necessarily know if you had the canker and then you didn't realize that that was a sexually transmitted disease. once you got into the secondary syphilis phase, you're going to have this rash that comes, then goes away, and so you may even think, if you're not aware of such diseases, that you had an allergic reaction. So the real problem comes in when we go from secondary to tertiary syphilis. So tertiary syphilis, we're moved past the fact that this bacteria is multiplying in the blood, and we're now having this bacteria multiply in organs, tissue sites, so it can result in paralysis, blindness, heart disease. So you can see here a gumma. So a gumma is a walled-off component of these spirochetes that our macrophages, our phagocytic cells, have tried to contain, and they've been unable to contain that very well, so, or get rid of this particular bacteria, and so they essentially wall it off. So I did like a little example here. These are all big macrophages, and you can see here there's, an infected set of tissue where there's bacteria in that area. And so that's the physiological manifestations of a gumma shown here. And so it's very deforming. These are gummas shown on various parts of a person's body. And then, of course, syphilis is one of the few diseases that can actually be diagnosed with an eye exam. So it causes an irregular pupil shape. So, as I mentioned earlier, it's not just the person who originally acquired syphilis that suffers, it is the entire family. So, children born of parents with syphilis results in failure to thrive. Failure to thrive is a definition that just means that this baby is not gaining weight, it's not, able to keep down food, and so it's just a complete loss of most of the infants that are born to parents of syphilis. Those that do not die end up having things like these Hutchinson's teeth. So Hutchinson's teeth are these kind of peg-shaped teeth, and so if they progress and live, to an older age, then, essentially they could have protected against these malformations that have taken place due to syphilis simply by the treatment of penicillin. This is from 2011, so it's a bit older data, but syphilis is still prevalent in Indiana. It's prevalent all across the world. And I find it interesting. So here is Marion County, where Marion University is located. And so you can see here that the congenital cases in 2002 were much, much higher than they are now. So we've pretty much reduced or removed most of the congenital cases. We see that the female cases in blue, those female cases have stayed at pretty much like a linear, not increasing, not decreasing significantly. But the male cases are still on the rise. And so my question for you is, why do we have a higher proportion of males suffering from syphilis when the females are not? So can you come up with some rationale as to why men would be more likely to be exposed? So typically when we're discussing this in class, a lot of students come up with the following possibilities, one of which is promiscuity. So promiscuity, person to person, so not using condoms, and of course promiscuity in homosexual relationships where condoms may not be needed to prevent pregnancy and things like that. And so promiscuity spreading the infection. The other idea is, in certain groups or cultures, men are more hesitant to seek out health care. And so if they're not seeking out health care, they don't realize that they're infected, and so they're propagating the disease. Borrelia. So Borrelia burgdorferi is the causative agent of Lyme disease. It is also a spirochete. It is transmitted through an arthropod vector. And so an arthropod vector in this case is a deer tick. And so when we think about the life cycle of Borrelia, I'm going to go into this brief bit of information. So you have a larval tick. This larval tick picks up Borrelia from a mouse, so its first blood meal. So the infected larval tick then develops into a nymph. At this point, the nymph can come in contact with humans, can come in contact with deer. And so humans are actually an accidental host. We are not intended to be part of the life cycle of this particular bacteria. And instead, it's commonly found in deer, and then of course back to the deer mouse found here. So Borrelia burgdorferi, when it causes an infection, most of the cases result in a bullseye rash, which is really handy because this is a highly indicative a feature that allows doctors to quickly diagnose. I want you to think for a moment, how then do we diagnose people who do not come in with a bullseye rash? Because it's a non-fatal disease, it's slowly progressive, and it's going to mimic neuromuscular and rheumatoid symptoms. So the difficulty of diagnosing this infectious agent is actually really, really hard if it is an atypical case where the bullseye rash is not present. And so that is one of the failures of our healthcare system, in the sense that we rely a lot on the highest proportion and we treat from there. So a person who does not exhibit the bullseye rash will probably have a bit of a delay in receiving proper health care. And so this can also be treated by very common antibiotics, tetracyclines and amoxicillin. The curviform bacteria, so when we move to curviform, so you can see that they're not quite as long as a spirochete, but they do have the bends and twists. The two curviform bacteria that we're going to discuss are that of Campylobacter and Helicobacter. Campylobacter has polar flagella, so it allows it to be a motile bacteria. It's typically in our GI tract, can be in the oral cavity of birds or mammals. The fact that it lies in the GI tract causes the transmission usually through the fecal-oral route. And so typically it is contaminated beverages and food that are going to cause the spread of Campylobacter jejuni. Once the bacteria is in the colon, it can burrow into the colon tissues, and it then starts secreting a heat label enterotoxin. This heat label enterotoxin results in a really uncomfortable secretory diarrhea, such as that of what you would find with cholera. And so the patient is going to suffer from pretty severe headache, abdominal pain, bloody, watery stools. But at this point, the last thing you would ever possibly want to do is give them things like Imodium or things to staunch the diarrhea, because if you were to do that, you're going to keep that toxin in. So the Campylobacter jejuni toxin, or CJT toxin, is what's responsible for the secretory diarrhea. So if you don't flush the bacteria out, you're keeping the toxin in, and the bacteria is going to continue to cause diarrhea. So instead, the therapy is simply give them sufficient water, electrolytes. Helicobacter pylori. So finding this particular bacteria in 1979 kind of has changed the way we think about ulcers and stomach issues. So 90% of all stomach ulcers are actually caused by H. pylori. In particular, I find it very interesting that people with type O blood actually have a nearly double incidence rate than that of people with AB or AB. And so I want you to reflect on, why do people with type O blood have a higher incidence? So to address that, let's take a look at blood typing. So when we look at type O blood, the type O blood has absolutely no proteins on their cell surface. Right? So you can see here they have no type A proteins, no type B proteins. And then as a consequence of that, right, it mimicking the type O blood, H. pylori has actually several reasons why it causes harm, and you can see those in the notes, so feel free to peruse those on the previous slide. The notes of this PowerPoint are located when you open up the digital file. It's at the bottom. And so one reason that H. pylori is causing harm in this sort of scenario is because the antigens of H. pylori actually mimic that of these blood types. And so our O blood is going to see that as foreign, so our O blood actually attacks H. pylori a bit more aggressively. And because of the attaching and the protein attachments that happen with H. pylori, patients who have type O blood are actually more permissible to the infection. So H. pylori can actually burrow deeper in those patients, which in turn causes duodenal ulcers. So the last three bacteria that we're going to discuss that are completely atypical. So these guys do not have a body shape that is similar to that of most of the bacteria that we've discussed at this point. And so Rickettsia, or Rickettsia rickettsii, for example, causes Rocky Mountain spotted fever. You can see here that they are obligate intracellular parasites. So here is a very, very large macrophage, and you can see that on the inside of the macrophage there are bacterial cells. These are gram-negative cell walls, but they're pleomorphic, meaning that they are rods or coccobacilli, so they have multiple shapes, which is why we don't throw them into the coccus or the bacillus lectures. They can transmit through various arthropods, can do fleas, lice, things like that. And of course, once they are in our bodies, they're going to cause vascular leakage, thrombosis. They're definitely getting into our vascular lining. And so Rickettsia rickettsii is transmitted through a tick bite. And so similar to that of Borrelia burgdorferi, tick bites are the arthropod vector. And so once we have the rickettsia within our bodies, we're going to have central nervous damage. We're going to have a rash. So the Rocky Mountain spotted fever is due to the rash that develops. And then, of course, we have various vascular damage because that's where this particular bacteria is going to burrow. So here is an image of a child that is suffering from Rocky Mountain spotted fever. The CNS is the fatal component of this particular infection. So 20% of all cases, if you are left completely untreated, it can lead to death. Chlamydia trachomatis is a STI. So a trachoma is when the bacteria is attaching to our mucal membrane, so eye, genitouria, and lungs. And so as a consequence, those are then the sites of infection. And so you can have an ocular trachoma. So an ocular trachoma is shown here, which can cause actually malformations and issues with the eye cavity and ultimately lead to blindness. You can also have conjunctivitis. So as the baby is born to a mother who has chlamydia, it can end up causing a complete covering of then the eye. So that's shown here, the conjunctivitis. So the more politically correct term is STI, that is a sexually transmitted infection, because it's not a disease since it is easily treated with that of antibiotics. So we are indeed the human reservoir, so transmission of these two strains. The trachoma is the mucous membranes of the eye, so when we have chlamydia with a trachoma, that is then damaging eyes, genitourinary tract, or lungs. Versus the lymphogranuloma venereum is shown here. So lymphogranuloma is a swollen lymph node, and so you can see that in both of these cases. And it causes a pretty severe disfigurement of the genital area. The final bacteria that we'll talk about in this lecture is that of a mycoplasma. So the most common human infection is that of Mycoplasma pneumoniae, and that is indeed walking pneumonia for those in common terms. It's community acquired, so it's picked up from person to person, spread through respiratory droplets and things like that. The final bit of information about bacteria, there are bacteria that cause dental disease, so the kind of fuzzy feeling that your teeth get at the end of a day, especially if you've had high quantities of sugar, that is a biofilm. And so the biofilm of our bacterial community on those teeth can actually cause some pretty severe damage. In particular, you have early species of Streptococcus, eventually followed by Actinomyces. They form a dental plaque, so if we don't properly floss, brush our teeth on a day-to-day basis or multiple times a day, these plaques, the dental plaques, end up causing a grippy area for additional bacteria to attach. And so the additional bacteria that come in are very common. Lactobacillus is consumed through various foods. And then, of course, bacteria, when they digest carbohydrates, produce acid and gas. So this acid and gas, as it's digested through the mouth, will cause tooth damage. And so periodontal disease is caused by the biofilms formed especially around the root area. So the root area is in very close association with that of the gums. And so the gingiva of the gums become inflamed due to the, so periodontal disease takes place, especially gingivitis takes place because of the dental plaques that are aligned there along the root. So when you're flossing, because flossing is actually the only way for you to get into that sulcus. And so when you floss, you need to get into each sulcus here kind of deep so that you can get rid of the dental plaque. And so those pockets between the teeth, when the dental hygienist goes in and they're poking your gums to see if they bleed, it's to make sure that you're not suffering from gingivitis. And then, of course, those pockets can be evaded by these dental plaques and communities of bacteria. And then if it continues to take place, you'll have the gums will bleed, and of course the bones are getting digested due to the acidic environment produced by the bacteria. So this is the conclusion of chapter 21. It's a relatively short chapter. For the most part, focus on the bacteria, the scientific name of the bacteria, the disease that is caused, and what is unique about it. What is your real take-home message? So in the sense of, periodontal disease, we know that unhealthy communities of bacteria in our mouths can lead to endocarditis and many issues with our entire body. So maintaining a healthy mouth actually ends up causing people to maintain a healthy heart. So hopefully everybody is going to go floss at the end of this lecture. Do reach out if you have questions or concerns.